Alright, so here we are with part 2 of chapter 1, and this is where we left off for part 1, looking at spontaneous generation versus biogenesis. And as I mentioned at the end, this really was both a scientific issue as well as religious issue. But a person named Louis Pasteur, which some of you may have heard about, he was very influential in this process of saying that spontaneous generation did not occur, that life came from life, specifically with microorganisms. And he did a lot of other work within microbiology. He started the process of using pasteurization to kill off yeast and prevented stored food from going bad, specifically with wine and that yeast component. And so pasteurization today is a major way that we prepare and keep our food safe, as well as available for somewhat long-term storage. He was also influential in developing the process of vaccines, and his work looked at creating vaccines against anthrax and rabies. And then he also hypothesized that air contained contaminating microbes. Louis Pasteur is famous with what are called the swan neck flask tests. All right, the swan neck flask experiments. And so here you have a broth in this flask, and it has this swan-shaped neck. All right, so the significance of this, the shape of this, is that the microbes were not necessarily in the air, so to speak, but rather they were in the dust, attached to the dust particles that were in the air. And so what he would do is he'd pour broth into a flask like this, and he would get it in through this neck, and so this area was open right here. And then he would heat this, and once he heated it, let it cool off, that heating process killed off the microorganisms, the bacteria, the yeast. And then he just let it sit. And after several hours or days, the broth remains pure indefinitely. It stays this nice transparent yellow color. Once you get into the lab, you'll see what a clean control broth looks like relative to a broth that is contaminated or specifically has the bacteria in it that you want it to grow. All right, so the issue is that with this end being open, air can get in through here, and so there was, one would think, well, you could just seal the end there and allow no air to get in. Well, the critics said that there were basically life spirits that flow through the air. So if you closed off the air here, then you closed off those life spirits. But this opening remained open, and when the air came in here and it had dust particles, the dust particles would settle in this area of the neck of the flask. Air could still travel up in this direction and maintain air current for this broth. And it remains pure. And this figure says hours to days. But in all reality, there were experiments of Pasteur's that went on for years. He had kind of a makeshift lab in his house, and it's an old house, but he actually had this lab underneath the staircase in his home, kind of like how Harry Potter had his room underneath the staircase in his aunt and uncle's house. And some of these broths would get put in that little room underneath the stairs and just left there. And years later, the broth remained clear. All right, so and then in scenario 2, The flask is tipped so that the broth makes contact with the dust particles that have settled in this area, and it gets tipped back, and then the broth is contaminated because the dust from here got in. The dust is dirty, and you had contamination. And then same process as over here, except over time, then you break the neck here and allow for all the dust and microbes to get down into the broth, and you have contaminated broth. So this was actually a very important experiment that was done that really changed the way that we thought about science and about microbiology. All right, then there was Robert Koch, who developed germ theory of disease, and this simply states that microbes cause infectious disease. Right? It's a germ that causes disease. And Robert Koch was important in identifying etiological agents of an infectious disease. So what causes the disease? His work primarily looked at anthrax. And we will look at Koch's postulates later, but just briefly here. Koch's postulates state, there's 4 of them, the same organism must be present in every case of the disease. And once that organism gets the disease, you have to, or organism or animal, I've got a picture here of a mouse. All right, here, the same organism must be present in every case of the disease, but not present in healthy individuals. So here you have the diseased mice, and you can identify these particular bacteria, but the healthy population, the healthy mice here, you can't find this pathogen. All right, then postulate 2, you have to be able to take the causative agent from this disease case and grow it in the lab, and be able to grow it as a pure culture so that you can say, indeed, this organism is causing this disease. So you've got the orange matching the orange here. All right, third postulate is that then you have to infect a healthy host, this gray mouse, with the bacteria, and you have to then get the same disease as you saw in the first round of disease. Fourth postulate is then you have to be able to re-isolate the same pathogen from this second exposure, second case of illness, and it is identical to what you isolated from the first. So this went a long way in that idea of germ theory, that germs cause disease. This was held onto for a very long time. Physicians, researchers, clinicians still hold Koch's postulates as legitimate scenarios, although we do know that there can be exceptions to this rule. All right, we know that HIV leads to the development of AIDS. All right, but in the real scenario, HIV, we can't pull it from a human. Right, we can pull HIV from a human patient. It can be isolated, but it stops here because ethically you can't reinfect a human with the virus and get the same result. I mean, you would get it. People have gotten into a lot of trouble with intentionally inoculating people with tainted blood. That sounds awful, but there have been cases in the news where that has happened. And so you can't do that. So there are a few exceptions. But still, Koch's postulates are very important to the field of microbiology. All right, then looking at morphology and physiology. All right, with the moving off of Koch's postulates, all right, here you have to be able to identify the bacteria, and you identify it partially on how it grows on this petri dish, what it looks like, and then also using the microscope and seeing what individual cells look like. All right, morphology is going to be the shape and size and arrangement of bacteria cells. So looking at the bacteria that grows on the surface of a petri dish, looking to see what shape it is, how big it is, how it interacts with its neighbor. Those what kind of seem like simple pieces of information can actually be very important in identifying an organism and therefore being able to identify a disease. So between the late 1800s and mid-1900s, most of the major bacterial pathogens known today were first characterized because in some cases the bacteria don't change a whole lot. All right, anthrax then is what anthrax is now. Staph, what that was then, is very similar to what you have now. Right, you do have the issues of antibiotic resistance. So in 1900, you didn't have MRSA. All right, MRSA is methicillin-resistant Staph aureus. Why didn't you have that? Well, because antibiotics didn't develop until the late 1930s, early 1940s. All right, so you can use morphology of the cells to help identify and therefore classify bacteria. And there are different ways of culture techniques, isolation, staining that came out of this learning process that are used today. All right, and actually that's where we're going to stop for part 2 because the next slide starts a pretty different section.